So there's those big megakaryocytes, but there's all sorts in here. And it's a mess really, unless you know what you're looking at. It's just chaos. But believe it or not, we've been able to sort of work out that, take that mess and work out that what we've got in the bone marrow is everything starts off as a stem cell. We have these very primitive early stem cells, okay? And they're not many of them. I'm going to talk about them in a minute. But stem cells sort of can grow up into these what we call progenitor cells, and there's two main progenitors, the lymphoid ones which go off and develop that immune system, and these are called the myeloid progenitor, which develops into these the other white blood cells, including the neutrophils and the platelets and the monocytes, things I've talked about, and the red blood cells we think is part of the myeloid system. So that's sort of, and I'll come back to that later. That's sort of how we think this mess is organised. Now, how are they produced by the bone marrow? And again, this is a simplified version of what I've just shown you, the stem cell with these two major sort of progenitor cells, and then we have these immature cells in the bone marrow which grow up, and these cells are the blood cells. And so in the bone marrow we have sort of cells dividing and amplifying, and in the blood we have the sort of mature blood cells which do useful work. And I'll come back to that. So how does it happen? And there's four concepts, and I'm going to labour these again, so don't worry if you don't get them. Stem cells are important. We have to have stem cells. And so if your stem cell is going to grow up and turn into a blood cell that dies, you're going to run out of them. So stem cells have to be able to renew themselves. So they have to generate cells which are going to turn into blood cells, but they also have to renew themselves. And it's very important. It's called self-renewal, because there's not many stem cells, okay? We have to have cells dividing. We have to proliferate. We've got to make, 200 billion red blood cells a day. Your bone marrow's got to have cells dividing rapidly and expanding, okay? That's very important. And we also have to have this is a concept called differentiation. Differentiation is growing up. So the cells have to change from being stem cells and immature cells into mature blood cells that are useful cells that do their job, okay? And finally, cells have to die. Cells are programmed to die, almost all of them. They have to die at a certain time. And that's set, it's almost set into their machinery that they die at a certain time. The only exception is we actually want some cells not to die too soon, and that's those immune memory cells. So if I get vaccinated against the flu, I don't want those cells to be dead in 3 weeks' time. I want them to be around next year and the year after potentially. Flu changes, so we have to get a new vaccine, but my tetanus vaccine I had 10 years ago, there's still cells around that remember that tetanus, and if I get tetanus in my bloodstream, they'll be there and they'll react to it immediately and get rid of it before it can cause harm. So we do need some cells to live longer. We need some cells to switch off that death. We call it apoptosis, which is sort of programmed death, okay? We want some cells not to do that, but most cells need to do that at some point in their lifespan, sort of the end of their lifespan really. Okay, I want to come back to stem cells. They're very rare, 1 in thousands. We've probably only got a few thousand stem cells in our bone marrow, and we've got trillions of cells in our bone marrow. So there's not many of them. And they don't come into the blood in adults. In newborn babies, they're present in the blood, but very rapidly they revert to the bone marrow. They come out into the bloodstream a bit after chemotherapy. If we give someone chemotherapy and their blood count is affected by that, as they recover, stem cells spill into the blood temporarily. And they also can come out into the blood under the influence of certain hormones or cytokines, particularly one called GCSF, and there'll be people in this room who know what I'm talking about in terms of that. So we often make use of that if we want to collect stem cells. We give people chemotherapy and then some injections of this GCSF stuff, and then we can harvest cells out of the bone, stem cells out of the blood, rather than having to get it out of the bone marrow. And they're so infrequent we don't actually know. They look pretty unremarkable, we think. And they don't divide very often. They don't need to. But when a stem cell divides, it's got to produce another stem cell, okay? If you don't, you've only got 2,000 or 3,000, that's not going to last your lifetime. So when a stem cell divides, it's got to produce another stem cell, but it's also got to produce a cell that started to mature, to grow up, a progenitor cell. And this picture shows you that. It's what we call asynchronous division. So when a stem cell divides, it doesn't produce two identical cells. It produces one that's exactly the same as what it was, and it produces another one which has changed. That's the self-renewal part. And it produced another one which has changed slightly, the progenitor cell. And that's the difference. So it's changed. It started to grow up. Okay, not much at that stage. So stem cells have to do that. If you just produced 2 more stem cells, well, that's not helpful, because that stem cell's not going to carry oxygen around your body. It's not going to fight infection or stop you bleeding to death. But if you just produce 2 progenitor cells, which are then going to grow up, that's no use, because you've only got a few thousand stem cells and you'll run out of them very quickly. Okay, so you've got to have that sort of this uneven division. And the progenitor cell then can go and proliferate and produce billions of cells to make mature cells as it's growing up. And then it's got to die, okay? So just this concept that we've got these two things happening in the bone marrow. Once the stem cells produce one of those cells that started to grow up, it's got to divide a lot and produce a lot more cells, but it's also got to grow up. So we have this proliferation. So, to get billions of cells a day, the cells have got to divide. They've got to divide like crazy, okay? So one cell goes to two cells goes to four, etc. And that sort of amplifies everything. But they've also got to grow up. A stem cell's actually useless in terms of doing anything useful in the bloodstream. Okay, so it's actually got to differentiate, and they've got to grow up into mature cells, which actually can do have important function out in the body. Okay, and then they die, the mature cells, and that's important. I'll come back to why that's important, because if cells don't die, we get into trouble as well. So what do we expect our bone marrow to do? We have to ensure the stem cells never run out, have to produce vast quantities of mature blood cells in just the right amounts every day, and we have to actually be able to change that. So if say I get pneumonia, the bone marrow has to produce a lot more white blood cells to fight that infection, okay? So it has to be able to respond to change. And the second thing, another example of that is, apart from producing the sort of neutrophils to fight the pneumonia, we also want to produce some immune cells which will remember that bug, so if I get it again, it will get it efficiently next time. And another example is if I donate a unit of blood, or if I get knocked off my bike and bleed, I have to produce lots of red blood cells to replace the ones I've lost, so much faster than usual. And I also have to produce platelets to help with that clotting process, because you use up platelets when you cut yourself every time. Okay? So how do they know when to do this? That's the problem. I mean, does it just happen routinely? I mean, how do they know when to divide? How do they know I've got an infection that I need to make more white blood cells? Well, it's because signals come from outside these cells, which are sort of going about their business. And signals bind to proteins. I've got a picture of this in a minute, so you don't need to necessarily understand this, the words here, but they bind to those sort of proteins on the surface of cells, which send signals into the cell, into the nucleus, to tell it to change. And it sends those signals to the nucleus, and what that does usually is to switch on certain genes or switch off certain genes, which makes cells change their behaviour. So that behaviour may be to divide more rapidly, stop dividing, get active, or to die. All those things are controlled by signals coming from outside the cell, binding to something on the surface of the cell, sending a signal to the nucleus, which changes how genes are expressed. And this is a picture of that. So there'll be an exam after this before anyone's allowed to leave the room. I mean, it's an incredibly complex process, okay? This is a simplified version. This is a cell, okay? And this is one of those, the funny hat with the receptors on the surface, okay? They tend to sit on the surface and they sort of span this membrane, okay? And what happens is a signal comes from outside, okay? Usually a little protein or a small molecule or something. Can be another cell binding to it, adjacent to it. That signal then changes something, sends a message to the nucleus, which then changes the gene expression, means it either stops producing certain proteins or produces different proteins, which means the cells change. And that might mean they start dividing, it might mean they start growing up, it might mean they become more active, or it might mean they die. And these signals do that. That's how we think this happens. And this is an example which we're familiar with. This is in developing red blood cells. There is a receptor which I'm going to show you again in a minute, for a stimulating hormone called erythropoietin, and it's controlled by this other protein called JAK2. And if JAK2 usually inhibits any signal being sent, but when the erythropoietin binds this receptor, JAK2 is sort of activated, and the signal goes to the cells to divide and make more red blood cells. If JAK2 has a mutation which activates it without this message, then you make too many red blood cells. And we think this underlies a disease called polycythemia vera, where we produce too many red blood cells. Okay, so you can see how a mutation in a gene can actually make a cell behave badly without the appropriate signals coming in. And so these signals, we think of them as cytokines. They're like hormones that come from outside the cell and give a signal to the cell to do something different. And they do it via receptors. And the best known one is for white blood cells, I guess, is the GCSF, or granulocyte colony stimulating factor. Okay, and that stimulates the bone marrow to produce more neutrophils, and it also makes the neutrophils that bind that signal to actually become more active as well. And the lymphocytes the immune system has this whole series of these cytokines called interleukins.